Now we are starting the part devoted to protein purification. What is chromatography? Chromatography is a method of separation and analysis of mixtures of substances, as well as the study of physiochemical properties of substances. The word chromatography is from the Greek word chroma, color, and graphene, to write. The name of the method is associated with the first chromatography experiments, during which the developer of the method separated brightly colored plant segments. Method is based on the distribution of substances between two phases, stationary and mobile. Liquid chromatography is a type of chromatography in which the mobile phase, the eluent, is a liquid. The stationary phase can be a solid sorbent, a solid carrier with a liquid or gel applied to its surface. In the column version, a portion of the separated mixture of substances is passed through a column filled with a stationary phase in an eluent stream that moves under pressure or gravity. On this slide, you can see several variants of columns. Also, you can see here how a narrow strip of a sample applied on the surface of the column moves down the column and components of the mixture begin to separate, and the separate bands of components begin to expand. If the strength of the interaction of the components of the mixture with the stationary phase is significantly different, then the components can split into separate bands. The output of the components from the column is monitored using a UV detector. The graph of the dependence of the detector's signal from the time or volume of the eluate is called a chromatogram. You can also see it on the slide. We had two substances, two colors, and two peaks in the end. The peaks on the chromatogram correspond to a separate band of each component. At the beginning, we had a mixture of two substances, and in the end, we got two separate, well-resolved peaks of two samples. There are two types of liquid chromatography: analytical and preparative chromatography. Analytical chromatography is used for the analysis of mixtures of substances or the development of purification schemes. Preparative chromatography is used to isolate pure components in large quantities by using the selected technology. What else should I mention talking about chromatography? It is resolution. On the previous slide, you saw well-resolved peaks. On this slide, you also can see such peaks, but it happens that substances are not well-resolved, and you get one wide peak. Such a situation is shown on the upper left graph. This peak contains two substances and two peaks, but they are situated so close to each other that such a system cannot separate them. In all fraction, you will have two substances, but in different proportions. In first fractions, you will have more substance 1 and less substance 2. And in last fractions, you will have more substance 2 and less substance 1. There is a term for characterization the degree of separation. It's resolution, RS. The resolution is good when RS is higher than 1. When the resolution is lower than 1, you will never see two distinct peaks after exit from a column. The resolution of two chromatographic peaks, RS, takes into account not only their locations, retention time of each peak, but also takes into account the widths of peaks, W1 and W2. When performing quantitative analysis, they tend to RS resolutions from 1.2 to 1.5 RS values greater or equal to 2 should be avoided due to the long duration of the analysis. Now, I'll tell you what types of liquid chromatography exist. The main types of liquid chromatography used for protein purification are ion exchange chromatography, hydrophobic chromatography, affinity chromatography, and gel filtration. Here you can see them on the picture. Let's focus on each in more detail. Let's start with an introduction to ion exchange chromatography. Ion exchange chromatography is often used as the first stage of protein purification. It allows to separate ions and polar molecules based on the charges of the separated molecules. This type of chromatography allows you to separate almost any charged molecules, including large, proteins, small, molecules of nucleotides and amino acids. In this type of chromatography, the stationary phase is a cross-linked polymer resin to which ionic functional groups are covalently bonded. The counter-ions to these fixed charges are mobile and can be displaced by ions that compete for more favorably or exchange sites. Ion exchange chromatography makes it possible to separate molecules based on ionic interactions. The stationary phase has charged functional groups that interact with the analyzed ionized molecules of the opposite charge. This variant of chromatography is classified into two types: anion and cation exchange chromatography. Anion exchange resin delays negatively charged anions, since the stationary phase has positively charged functional groups. Cation exchange resin delays positively charged cations, since the stationary phase has negatively charged functional groups. Ion exchange resins are divided into four groups: strong acid cation exchangers, weak acid cation exchangers, strong basic anion exchangers, and weak basic anion exchangers. On this slide, you can see variants of such resins. For example, weak anion exchanger is DEAE cepharos, strong anion exchanger Q cepharos, weak cation exchanger CM cepharos, and strong cation exchanger is SP cepharos. What convenience of one or another type of chromatography? We can find conditions when target protein will stay bonded to the sorbent longer than other proteins. Or it can be more useful to use opposite condition when target protein doesn't bind to resin but all other cell proteins bind. The next type of chromatography is hydrophobic chromatography. In hydrophobic chromatography, the retention of separable substances is due to hydrophobic interaction of their molecules with a relatively nonpolar stationary phase. And to change the elution force of the mobile phase, first of all, a change in the ionic strength, salt concentration in the mobile phase, is used. Resins in hydrophobic chromatography can be divided into two groups, depending on their interaction with the sample. Resins with direct alkyl chains, butyl, octyl, ethyl, and isopropyl, showing a purely hydrophobic character, and resins with aryl ligands, phenyl, have mixed properties, aromatic and hydrophobic interactions, as well as the absence of a charge, plays a role in the final chromatographic properties. If the target protein does not bind at a high salt concentration, a more hydrophobic resin should be used. If the target protein binds to the resin so strongly that nonpolar additives are needed to elute it from the resin, it is necessary to lower the salt concentration in the starting buffer or use a less hydrophobic resin. So using difference in hydrophobicity of a target protein and other cellular proteins, we can purify one from others. The next type of chromatography is affine chromatography. It's a very convenient type of chromatography. The main feature that determines the high efficiency or affinity chromatography is the separation is based not on the difference in the physiochemical characteristics of the molecule, charge, shape, and size, but on the specific functional properties that distinguish this protein from many other biopolymers. The ligands are compounds whose interaction with the separated substances is based on the biological functions. Thus, when separating enzymes, their substrates, inhibitors, or coenzymes serve as ligands, or antigens. It is antibodies. The stationary phase in affine chromatography is a specially obtained sorbent, usually constructed according to the scheme carrier, connecting link, leg, specific ligand. The carrier is most often cepharos or agarose derivatives having crosslinks. The addition of a ligand or leg to it, containing as a rule an amino group, is carried out after activation of cepharos with bromcyan. Such carriers for affinity chromatography can be obtained by yourself in the laboratory, or you can buy commercial affine resins. There are two more types of sorbents. The first one is metal chelated affinity chromatography, sorbent with immobilized metal ions such as nickel, cobalt, zinc, and copper. This type of affinity chromatography is based on the interaction of individual amino acid residues in proteins, histidines, cysteines, and partially tryptophans. With transition metal cations, these resins are most often used to purify recombinant proteins containing a histidine tag. His tag is coded in plasmid, so you already know the purification scheme on the step of choosing plasmid vector for cloning. The second one is heparin cephalose. It is used to purify plasma proteins, as well as DNA, RNA binding proteins. Such proteins bind heparin, while other cell proteins don't. One more type of chromatography that should be also mentioned in our lecture is gel filtration. It's the final in a series of chromatographic purification. Gel filtration or size exclusion chromatography is a type of chromatography in which the molecules of sample are separated by size due to their different ability to penetrate into the pores of the stationary phase. The main rule of this type of chromatography is as smaller amount of your sample and the longer it weighs, length of a column, then higher resolution. Unlike all the chromatographs described above, during gel filtration, the stationary phase remains chemically inert and does not interact with the separated substances. Sorbent has pores of different sizes. On the slide you can see schematically view. The largest molecules capable of penetrating into the minimum number of pores of the stationary phase are the first to leave the column. The last molecules, with small sizes that freely penetrate into the pores, come out. So high molecular substances pass between the granules, so their retention volume is equal to the volume of the column minus the volume of the stationary phase, the so-called free volume. They elute first. Medium-sized molecules are placed in the pores of the sorbent, but not completely. Therefore, their retention volume is slightly higher than the free volume. They are eluted second. The smallest molecules freely enter the pores together with the solvent molecules. Therefore, their retention volume in the column is much higher than the free one and approaches the total volume of the column. They are eluted last. On this slide, I show you an example of separation of mixture of proteins using this type of chromatography. On the exit, we have number of peaks corresponding to individual proteins. For each branded gel filtration column, the manufacturer provides a standard chromatogram, which allows you to navigate in what volume, at what minute a protein of a particular molecular weight comes out. In ideal, it's better to calibrate your column by yourself, because such a chromatogram will be more accurate. But for approximate orientation, you can also use a branded one. As you can see, the left and right chromatograms are a little bit different, both performed on SuperDex 75. But in the second case, the granules are modified and contain smaller and more rigid granules and narrower pores, and therefore the result of separation in it is much better. You should remember about such properties when choosing columns for purification. What else I wanted to tell in context of protein purification? Let's go back a little bit. By the time after the destruction of the cells, when we have realized that our target protein was in an insoluble form, we can try to isolate the protein from the debris. I'll tell you what to do with such an insoluble protein. First of all, treat the cellular debris with a denaturing agent, with urea. Urea helps the target protein to unfold and pass into a soluble form. On the first picture, you can see that globular protein unfolds into unstructured state. After that, you can use any of the mentioned above types chromatographic purification. Which one to choose? Use usual principles. If a recombinant protein has a histidine tag, then it is easiest to use metal affinity chromatography for purification. It is convenient because the renaturation of the recombinant protein can be carried out in the sorbent-bound state. The renaturation process should be as slow as possible, so that the target protein does not precipitate again. The last test that should be carried out after renaturation is the checking of the isolated protein activity. It will show if protein retains its structure and properties. Another way to check your protein is to use gel filtration. It will show you if the target protein is in monomeric or aggregate form. Moreover, set up a functional test. Here we should mention that gel filtration is not only a purification method, it can be used for characterization of the molecules. It can show the presence of dimers or multimeters aggregates in samples. It is also possible to conduct binding experiments and visually see complexes formation by the appearance of new peaks and the disappearance of previous peaks in comparison with the chromatograms of individual components. So I'll emphasize once more. If your target protein was in insoluble fraction, in cell debris, you can try to purify it in denaturate condition and renaturate it. And if you've got it pure in active monomer or dimer form, it's time to move to crystallization procedure.